All right, I got a full screen of people on my end, so I will start by welcoming everyone. So anyone that's attending this call for the first time, we've been holding this industry-wide call every month for really any company that wants to participate in the cultivated meat value chain. And the purpose of these calls is to introduce and welcome new solutions providers into the industry, ultimately with the goal of establishing new collaborations that can accelerate R&D and commercialization. So if your group is interested in presenting on a future call, or maybe you're working with a company that could benefit from being on these calls, please do feel free to put them in touch with me directly, and I'd be happy to work with them. So a few housekeeping items from our side before we start. I'll turn it over to Laura Braden from our policy team for an update.
Thanks, Elliot, and hi, everybody. Yeah, I just have a couple of quick updates I wanted to make everyone aware of. One is that there are two new funding opportunities that were recently announced by the Department of Energy and the Department of Defense that are relevant to alternative protein companies. The first, I'll post information here in the chat about the Department of Energy funding opportunity announcement that was recently made to award several proposals in the food and beverage industry of up to $7 million each that will advance sector-specific technology solutions to address challenges unique to the food and beverage industry. And the announcement specifically highlights alternative proteins as an area of interest. That link I posted in the chat is where the announcement itself and additional information can be downloaded. Then secondly, the Department of Defense announced a new funding opportunity for US companies, including alternative protein companies, to apply for biomanufacturing production facility grants. Food was highlighted as one of 5 defensive priority areas in this announcement, and the department expects to award 30 proposals with $2 million each to produce a business and technical plan. And more information, again, can be found at the link in the chat. And then finally, earlier this month, in a publication of the Unified Agenda for Federal Regulatory Agencies in the US, the USDA FSIS announced that it expects to publish its anticipated proposed rule on the labeling of cultivated meat in May of this year. So we'll be keeping a close eye out for that in a few months. Thanks.
Thank you, Laura. Yeah, definitely encourage US companies to take a look at those grant opportunities. Just a few other things from me before we begin. Last year, we surveyed the industry about scale-up and bioprocessing, and now we have the results compiled and ready to present at a webinar that's coming up on March 20th. So I'll drop a link to the event page and registration. There'll be an accompanying report with a ton of great information in it, so encourage you to keep an eye out for that and attend the webinar. Lastly, on March 26th, we're hosting another session of Fish and Chips, which is an event series that provides cultivated seafood scientists and innovators an opportunity to get together in smaller groups and talk through some of the challenges that they're specifically facing. So the topics for the March event will be omics, media optimization, and supply chains. And so if you're working on cultivated seafood and you want to connect with others in the field, join us for one of the 2 sessions on March 26th, and we'll drop those links in the chat. Alrighty, so today we are pleased to welcome Manish Gore from HiMedia Laboratories, who is going to be sharing some details about their end-to-end solutions for the cultivated meat industry. And then we will turn it over to Payar Radfar from Smart MCs, who will be discussing some of their microcarrier technology. I just want to note that Payar's talk is pre-recorded due to his location in Australia. And besides that, feel free to drop questions in the chat throughout, and we'll get to them at the end. Manish, I will turn it over to you.
Yeah, thank you so much. And thanks to the GFI team and everybody who have joined for this seminar. It is great to be here and meet all of you. So thank you so much, Elliot, Rennie, and the entire GFI team. So myself, Dr. Manish Gore, I'm from HiMedia Laboratories Pvt Limited, India. And I'll be taking you through our capabilities as an end-to-end solution provider to the cultivated meat industry. So to start, so I'll be discussing about our capabilities as an end-to-end solution provider to the cultivated meat industry. To start with, HiMedia is a bioscience company based in India, and it has served the humankind by developing molecular biology products, cell biology products, and starting with the microbiology products over 50 years. So we have been in this business for last 50 years, and we are among the top 3 global powdered media manufacturers. We have more than 30,000 SKUs, and we have the culture media capabilities, about 60 tons of media we can produce per day. So this is our production capacity. Coming to the global presence, we have nearly 16 manufacturing locations which are spread across India and abroad. So we also have now offices and warehouses based in India, Germany, and USA. So we can get in touch with you from different corners of the world and help you out. Our products are exported to more than 150 countries worldwide, and we have more than 335 global channel partners. So that is an extended reach across the entire globe. Coming to the another perspective of the global presence, we hold more than 4 international patents, and we are among the top 8 cell culture companies globally. More than 2,000 powder media can be manufactured or are manufactured by us per year, and we hold a huge number of citations in the Google Scholar and such research databases. And so far, we have been awarded with many national and international honors, as well as the grants, research grants to support our R&D and production scale research. Coming to the business verticals, HiMedia basically started as a microbiology company over 50 years back, and now it has extended its domain to molecular biology, cell biology, that is animal cell culture, and we have also moved into the plant tissue culture, hydroponics area. We are one of the providers of the chemicals and biochemicals to the cultivated meat industry as well, and the consumables and equipment. So we have been serving to different sectors of the biosciences and biotech companies, and cultivated meat is one of the very prominent sectors here. This is our recent headquarters, which is situated at Thane, which is near Mumbai, India. So this is our head office. Our production plant is located a few kilometers away from our headquarters. So this is our headquarters location. So this is our production plant where we have our media manufacturing facilities, R&D facilities. We are one of the largest producers of protein hydrolysates. So we even have the production plant or manufacturing plant for it. So coming to the cell biology division, we have different product ranges under the cell biology division, starting with media, viral vaccine platforms, bioproduction platform, that is biopharma, cell culture tested chemicals, cell analysis solutions, stem cells and primary cells. We have a huge library of human stem cells and primary cells, and are also currently working on animal cells as well. We provide different consumables, filtration solutions, and advanced instruments to the biotech and biopharma industries. So coming to the services module, we are the approved ATCC distributors in India, and we also provide custom services for cell culture and other biotech applications. We also organize skill development programs to enrich the young minds and help them understand the basics of the cell culture, cell biology, and related bioscience disciplines. So our capabilities, to start with that, I'll first describe our capabilities or our strengths in the case of media production. As we know, cell culture media is one of the most crucial raw material for cultivated meat industry, and we are at the forefront of the product of the cell culture media providers to the cultivated meat companies. We can produce nearly 60 tons of media per day at present, and this capacity is going to rise up to nearly 100 tons per day in the next 1 to 2 years. We also have advanced technologies and customization, which we bring in our cell culture media portfolio, and we have a bulk packing capacity in addition to the manufacturing capabilities as well. So in the case of powder media, we can yearly produce 10,000 kg of powder media per lot. And in the case of liquid cell culture media, we can produce up to nearly 2,000 liters per batch. And we have an automated filling line, which is ISO class 100 for liquid media. So as the production is important, the same way the quality checkpoints are very, very crucial when it comes to cell culture media. They start from testing our raw materials which go in our media, where we subject the raw materials to physicochemical testing, biological testing, analytical testing, and now even the food contaminant testing, which we are starting off for the food grade media. So while the media is being produced, we do a lot of in-process quality controls, where we test its uniformity, quality. As a result, the media that goes to our customers is as accurate to its technical data sheet, and you can expect the wonderful responses in your cell bioassays. Coming to the finished product testing, which is our final media, we subject it to the principal component analysis, where we again ensure the uniformity of the product content uniformity, and different physicochemical biological testings, and now even the food contaminant testing, which is very, very crucial for the cultivated meat industry. Our regulatory compliances, we have a wide, what I can say, our coverage with respect to the regulatory compliances. We have a QA and regulatory team who are in constant engagement and who are developing and getting the certifications in different areas. With respect to the cultivated meat industry, we have ISO 22000 certification for our production facility, as well as our quality testing facilities. So this ensures that the media, the chemicals, and all the products that we give are regulatory compliant, and we are happy to help you with regulatory submissions as well. So this is our so far journey when it comes to the cell biology, how it evolved in our department. We started off with primary cells, stem cells, vaccine industry, biopharma. Then in the case of COVID scenario, we were in direct communication with our Indian government, where we gave the COVID kits, and even the virus inactivation medium, viral transport medium, which attracted a huge interest in India. Since 2021, we are working in the cultivated meat domain and are currently engaged with more than 20 companies. We have recently submitted the food master files for our cultivated meat media to Singapore Food Agency, food food standards of Australia, New Zealand, that the regulatory body, and even the US FDA. We have also we have also started off with the 3D cell culture and 3D bioprinting domain as well. As we know, the importance of the cultivated meat, the climatic benefits, the animal, the cruelty-free aspect of the cultivated meat, and even the green environment aspect, the food security, food sustainability aspects, and the benefits associated with cultivated meat. So all the participants in today's seminar are very well aware about the benefits of the cultivated meat. So as everyone is aware, this is how the cultivated meat is produced in different companies, starting with this biopsy of the animal tissue, and then developing the primary cells, cell lines, growing them into the bioreactors. And you can either have an unstructured meat, or you can put it, mix it with a scaffold, bioprint it, and finally what you get is a structured meat at the end. So now we will be explaining you where do we fit into this entire process. As we are aware, there are a lot of gaps in the cultivated meat industry at present. And starting off with the cell line development, it is very important that the cell lines which are used for the cultivated meat, the availability is a major gap that we see in the present industry. Another major gap that we see, which we are at the forefront of fulfilling it, is a cell culture media. Customization is one very key aspect. Then the volume, that is the manufacturing volumes that the cultivated meat industries require. It is another important aspect. Then next is the turnaround time. How quickly we can provide the media to the customers is very, very crucial over here in the cultivated meat industry. Price is a major pain point when it comes to the cultivated meat media or cell culture media as such. As we know, the price of the media is one of the major contributors to the meat cost. We are also working now on the food grade and food safe media development. So this is another very crucial aspect for the cultivated meat industry, or a major gap that we see. And ultimately, above all, the regulatory compliance. If we come into the bioprocessing domain, the upstream bioprocess which is required for cultivated meat development it should be useful for getting a very high biomass. And that is how efficient the bioprocess should be when it comes to the cultivated meat. And finally, the 3D aspect part, 3D cell culture aspect, where the scaffolds that we use for developing the cultivated meat, they should be edible. The price point is also very crucial when it comes to 3D scaffolds, and they should support the development of biomass, so that the biomass which is developed in the bioreactors, the per liter biomass is very huge in the quantity. So we fit into these different domains in the cultivated meat, starting from providing the cells, like developing the cell line. Another is the cell culture media, which is one of the our USPs. Then even we provide the bioprocessing services to the cultivated meat industry. Having launched into the 3D cell culture domain, we also develop the scaffolds, provide bioprinters. So this is how we cover the entire aspect of the cultivated meat industry, and this is how we aim to be end-to-end solution providers. So how does our cultivated meat platform looks like? Our major platform giver is a culture media. So under the culture media, we provide powder media, liquid media, and now we can also provide the food grade media. So we are working on the developing the food grade media. Apart from the usual classical media like DMEM, MEM, RPMI, and so on, we can also provide a serum-free media, which is animal cell line specific media. Maybe it may be a avian species, or like a bird species, or maybe bovine species or equine species or any species. So we can provide a serum-free media for specific cell types as well. And ultimately, as I mentioned to you earlier, customization is our prime USP, as we understand considering the financial constraints and considering the innovation aspects and the huge requirements. Customization part has become a very dynamic, what I can say, crucial aspect in the cultivated meat for the cultivated meat companies, and we are open to customizing like minute components, their concentrations. We can help you with the optimization of those components and so on. Coming to the next platform, which is food grade chemicals and reagents. So we have a comprehensive range of cell culture grade chemicals, and most of them are, or all of them, I would say, are multi-compendial grade. That is, they meet the pharmacopoeial requirements. That is how the premium and pure they are. Now we can also provide the food grade chemicals, which is very important aspect for the cultivated meat industries. And under that, different reagents, different buffers that we can give to the cultivated meat companies. Coming to the service aspect, as I mentioned earlier, we can engage with you in development of primary cells, stem cells, and cell lines, and ultimately help you in optimization of media, fine-tuning the components, as well as the bioprocessing, that is the manufacturing aspect of the cultivated meat. And ultimately the use of edible scaffolds, 3D bioprinter, and making it more bioengineered cultivated meat, as like few of the companies that are working with bioprinting, like Stakeholder Foods. So this has become one of the crucial innovation aspect in the cultivated meat. So we are also at the forefront of it. Above all this, we also do evaluate the food safety aspect of the each media ingredient that becomes a part of the cultivated meat media. And we do the food safety evaluation as per the guidelines or the regulations, or I would say the requirements of SFA, FSANS, and even US FDA. We can also extend to European requirements or any particular country specific requirements. So we're open to have and engage in discussions with all the customers. So starting with one of the major platform, which is animal cell line platform. Here we can provide different types of the cells, services, and this is how we aim to be an end-to-end provider. Cell line security is very, very crucial aspect when any customer wants to share their cell line with us, or even for maybe, let's say, quality testing aspect, media development aspect. So we honor your intellectual property, and it will be intact. Your intellectual property and all those confidentiality concerns will be intact with us, and we follow all the necessary guidelines to make it work with you. To have the cell line development, we have our molecular cloning facility, which is very fascinating and which has been come up. And we have a lot of instruments under this molecular cloning facilities, starting from clone imager, single cell dispenser, transfection system, and even the recently one that we got is a seahorse system. And this is how we extend our domains and capabilities for molecular cloning that is required for cell line development. And this is our capabilities and what instruments do we have under other set of instruments in our molecular cloning facility. Cell culture media is another crucial platform and very important platform. I would say we are one of the biggest cell culture media suppliers to cultivated meat industry today. So currently that the media that we supply to you are cell culture grade, and the food grade media development is in progress. Here we are checking the food safety aspect of the media that we will be providing to you by qualifying each raw material, developing the media, testing it to make it that it is sure for use in the cultivated meat development. So customization, as I mentioned to you, is that we expect this is how these are a few of the test parameters that we do for our media. And now we are also developing the food contaminant testing profiles, where we will be testing for the testing the raw materials for allergens, pesticides, and we will ensure that they are free of food contaminants, and the media that we provide to you is food safe. So in the case of R&D, that is a media development aspect, here I would say that we can engage with you, and we have a high throughput tools facilities in our at our location, at our headquarters. And we have DOE capabilities. We can develop the media, we can develop the feed process, and not only develop the process but also validate it by quality by design approaches. With the help of automated platforms, we can make it really quick and high throughput, so that the turnaround time is as short as possible. So this is how you. So this is like an overall tentative outline that how do we go ahead with the media optimization. So we do it at different stages, where we first of all segregate the individual nutrient groups and understand which nutrient group is very crucial or component is very crucial. And once that is identified, we fine tune its concentration. So this is how we design the media. And next, coming to the feed, we also do it with the help of DOE approach, that is design of experiments approach. We have high level softwares and tools in our at our capabilities, and we use them at the best possible way, in a best possible manner, to design the effective medium for you. And it is tested comprehensively from the analytical part, physicochemical part, biological part, and now even the food safety part. So, and once that entire process of media development is set up, it is and optimized, it is also validated. So that the biomass that you give, like let's say a few hundreds of grams per liter, and so that we can so that the productivity of a medium will be very huge when we give it to you. To give an example, this is our STEM-in-1 media, which is a serum-free and xeno-free medium that we have developed for human MSCs. We can also provide a sample of this medium to you at present, and you can test it and let us know that if it works for you for maybe different animal species, or the mesenchymal stem cells of different animal species. And we can also engage and develop cell line specific or animal specific MSC media with you. So with respect to the product performance, we have tested it with the growth of the cells and the yield densities that we obtain, the morphology, even the characterization like osteogenic potential, adipogenic potential, chondrogenic potential, and any other like differentiation potentials that we evaluate for our stem cells. Coming to the next platform, which I mentioned to you, that is chemicals and reagents. So under this, we provide cell culture grade, multi-compendial grade, that is pharmacopoeial grade chemicals, as well as these chemicals are animal origin free. Currently, we are in the process of qualifying the food grade chemicals that we can provide to you in huge volumes. So these are the different chemicals, reagents, and what I can say, the requirements that go into the media, and we can provide those to you. So you can just have a look, and we are happy to engage with you in the further discussions. So this is our new CGMP facility, which is coming up as per the industry standard 5. So this will be developed with a QBD approach, and it is integrated with the PAC tools, which is very important when it comes to US FDA compliant facility development. It has data analytics incorporated into it, and as this CGMP facility is specifically developed for cultivated meat industry. We have already a huge manufacturing facility at our production site, but yet we are developing another facility only specifically for cultivated meat, and that is how we are serious about the cultivated meat, and we want to enable this industry to grow. These are the few of the pictures from our facility, that is a testing facility. This is how we store the chemicals. There is a separate facility for media development or media preparation. We have a cell culture facility, primary and stem cell culture facility, which is separate from our other cell culture facilities. We have more than 6 robotic liquid handling systems where we can conduct high throughput media development or media testing, and you can this is very huge and important platform for media development or media optimization. So these are the different sets of instruments when it comes to R&D, QC, upstream processing, and these are capabilities starting from, let's say, Citation 5 imaging reader. It is a multi-palette reader come the imaging platform. We have also the flow cytometer in our facility where we characterize our primary cells, stem cells, and cell lines for marker expression. We also, as I mentioned to you, we use DOE for media optimization. We also have upstream processing equipment in our facility, like starting from shaker incubators, like different liquid handling systems. We have Beckman Coulter systems, which which is useful for the high throughput screening. Describing about the bioreactor platforms, we have a huge bioprocessing facility, which have bioreactors of different capacities, starting from 1.5 liters to 5 liters to 10 liters, now even 50 liters. We also have a perfusion bioreactor at our facility, and that is how we can engage with you in bioprocessing services. So this is our 1.5 liter bioreactor facility. This is our 4 liter bioreactor facility. Then 5 liter bioreactors. These are our 10 liter bioreactors from Sartorius. So we have purchased these bioreactors of like one of the reputed manufacturers, and we also recently have this twin 50 liter bioreactors in our facility. So there are 2 sets of such 50 liter bioreactors, and we use it for biopharma, vaccine, and cultivated meat industry. As I mentioned to you, we also have the bioreactor, perfusion bioreactor capabilities as well. So this is our usual process flow for media development that we have. First of all, if we want to optimize any media for any of our cultivated meat clients, we usually start with the high throughput screening, which we conduct in the case of bioreactor tubes, let's say, and we use our high throughput platforms, or as I mentioned to you, we have robotic liquid handling systems. We use those kind of platforms for high throughput screening. Let's say if we have more than 1,000 media combinations that we have shortlisted by design of experiment software, we will be screening them with the help of our high throughput tools. Then next stage, we go to the lab scale stage where we use shake flask studies. Again, we screen our media combinations from it. Then we go to the lab scale bioreactors, which are ranging from, let's say, 0.4 liter to 1 liter. And we test the productivity of a media for the development or cultivating the biomass of the cells. As we know, for the cultivated meat industry, biomass is very important. The growth of the cells, the bulk mass of the cell is very important. And finally, the process optimization that we carry out in the 10 liter reactors. This will help us to validate what we have developed so far at lab scale. And ultimately, we move into pilot scale reactor, that is bioreactor of 50 liters, to validate the media combinations before providing them to you. Apart from this, we have a huge range of analytical services, and this is a very crucial domain that we see and very important for all of our cultivated meat grants. Starting from, we have HPLC, UHPLC. I will be shortly showing you those pictures as well. ICPMS, LCMS, HRMS, and we can do, let's say, spent media analysis. We can do any particular raw material analysis, your media analysis, maybe metabolite analysis, and so on. So this is the huge range of services that we open for cultivated meat industries. Coming to the capabilities in this sector, as we see, we have NOVA biochemical analyzer, which is useful for metabolite analysis, cell metabolite analysis. Then we have gel dock systems, chromatography systems, amino acid analyzer. We use it for ensuring the uniformity of the cell culture media that we provide to you. We have HPSs. We have a huge number of HPSs in our facility. Apart from that, we have UHPLC, LCMS, we have ICPMS for elemental analysis with the digester. We have capillary electrophoresis systems. Recently, we have got Lumos HRMS, Orbitrap Lumos HRMS. With the help of this system, we can analyze each and everything that you may require for your product. We also have FT-NIRs, which are installed in our production site. These are very crucial for monitoring of our cell culture media while it is being manufactured or produced at our production site. We have multi-purpose analyzers, we have Raman spectrophotometers. So we have end-to-end capabilities when it comes to advanced analytics. So these are some pictures for you, starting with our NOVA biochemical analyzers. So this is our amino acid analyzer. This is our NGC system, which is a downstream system. Protein analytics systems. We have UHPLC, LCMS, ICPMS, capillary electrophoresis systems. We have also started with our 3D cell culture facility, which will cater to 3D bioprinting, organoids, spheroids, and this is how we want that academia, industry, clinicians, all the sectors together to work, and in case of cultivated meat as well. Under the 3D cell culture facility, we have a 3D bioprinter, which is a 6-head bioprinter designed in India and developed in India. This is our specialized 3D incubator come bioreactor for organoid and spheroid development. In this facility, we have confocal microscope, rheometer for bioink or bioscaffold developed characterization, texture analyzer, which will be useful for analyzing your meat products. These are the different systems, like say freeze dryer. So currently we are engaging into contract research services for lyophilized products for cultivated meat industry. So this is our 3D cell culture capabilities. Under these capabilities, we can provide you with the edible scaffolds, 3D bioprinters, and even the organoid and spheroid models and the media for them. So this is our 3D bioprinters that we are selling across the globe, and we will be also happy to get in discussions for. It is a completely customizable model. So you tell us our requirements, and we will be providing the bioprinters as per your requirements. So ultimately, this is how we close the entire story, starting with if you want to use the same bioprocessing service for a cultivated meat. Like this is the usual media process flow. We will just be adding a 3D bioprinting part into this entire process flow, and to ensure that we can also engage with you in the meat development, meat characterization. Again, this is our pilot scale facility. That is a 50-liter bioreactor facility. So coming to the end of my presentation, this is how we offer end-to-end solutions, starting from products and instrumentation. Starting from media, chemicals, 3D culture scaffolds, bioprinter, and bioreactors. Even in the case of services, cell line development services, media development, upstream processes, 3D culture services. We are open to R&D collaborations, and we are working with many cultivated meat companies at present, where we are engaging with them in R&D collaborative projects. and the support. So we have a huge technical team, regulatory team who can help you with the regulatory compliances and evaluating the food safety of your media and helping you with the regulatory submissions. So this is our team at HiMedia. Our director, Dr. Vishal Wake, is a director in the cell biology domain. Our global business head, Dr. Rahul Joshi, Mr. Rahul Pant, who is a senior business specialist, Dr. Chayan Bhattacharjee, who is a field application scientist, and myself, Dr. Manish, research scientist at HiMedia. We also have our backend team who help us in providing different services to you. So thank you so much, and I'll be happy to answer any questions that you may have.
Thanks so much, Manish, for that presentation and all of those services that you offer. Great. Well, thank you so much. There's some other questions in the chat, but in the interest of time, Manish, maybe you could type out answers to those. And Renee, you can begin the next presentation. Thank you.
Thank you so much. Thanks
Hello.
to GFI for this opportunity.
I'm the CEO, co-founder of the SmartMCs, and today I'm going to present the range of products we've got for cultured meat, space particularly, and bioprocessing field. First of all, I gotta apologize first for making this a pre-recorded video and presentation because simply it was 3 a.m. in Australia and the time difference is really hard to schedule the talk. However, if you had any questions, I will leave our contact details at the end of this presentation and we can have a deep conversation about any questions that you might have. So SmartMCs came together with the idea of scaling cultivated meat from grams of cells to tons of meat. And the whole aim was actually to be able to enable this scale up without sacrificing things like texture of the meat or any of the taste. The company originally started with a group of researchers here in Sydney back in 2022. And a few years prior to that, we were working together and thinking about different solutions, including microcarriers, which I will talk more in this presentation about. But the whole story really was about how we can actually help the scale up of bioprocessing field. And we were officially founded in 2022. Our first product was launched a year later, 2023, and since then we have acquired over 25 clients in 3 different sectors of cultured meat, regenerative medicine, and vaccine production. Our business model is direct sales to clients, where we have directly provide them with biomaterials and macro solutions. We provide them with services including custom designing their products and some consultancy projects. And we also do R&D with actually different companies to use our intellectual properties across different fields for different applications. The list of our clients, partners, and collaborators are really a bit longer, but for the sake of their privacy, we have only included a few of them here. And as you can kind of see, we are scattered across different sectors, and we've been working with different companies to actually enable our venture to be able to produce the microcarriers, biomaterials, and other solutions for the field. So in that sense, our products divide into 3 main sectors. The first part is the smart microcarriers, which will be the kind of the heart of the company, and that's what the SmartMCs stand for. It's also then supported by smart hydrogels, which will be used for tissue mimicking. And the third category is the microfluidic solutions that is to enhance this research and the scale up of the bioprocessing. So I will be talking more and doing a deep dive into each of these and provide you with some micro context on that. And let's start first with the microcarriers. So lab-grown meat workflow, probably most of you have been familiarized with, often starts with taking a small biopsy from the animal and making a cell bank in the lab at a small scale where you actually isolate cell type of interest and start expanding them at a small scale where you can bank them or start directly the process of culturing. In the second step, often large scale cell expansion comes in, where cells actually get to expand at large scale. And here for adherent cells, cells that require a support structure, we will need to use microcarriers. In very simple terms, microcarriers are small spherical particles around 150 micron in size. So you can think about as thin as the human hair or a bit thicker. And the idea is they provide the support structure for cells to attach and grow on. And I will talk more about it in the next slides. But essentially, any cells like your muscle cells that they need to be static and stationary, or what we call them as adherent cells, they require this supportive structure, and these can be these little spherical balls that cells can attach to them, and then you can suspend them inside bioreactors. After you grow them to the number of cells you want, you can often go through a tissue maturation. You can differentiate these cells and scaffold them in the texture that you want. And the last stage often is you do the food, you process them and then package them into the final product. A similar case is actually for cell therapy, where you actually collect and establish cell lines, you expand them in large scale, and then you take them for a variety of downstream applications. But essentially, they are very common with cultured meat in the sense that you need large amount of cells. The difference can be around that in the cell therapy space, you're more caring about the quality of cell. Maybe in the cultured meat, you're less caring about that, and you're more caring about the mass number. But still, they share a lot of common knowledge in between them, and these are the cases that we kind of always have to go through. And if you were thinking about now the need at large scale, so to put it in the perspective, especially for the culturing space, you can see the list of different technologies here that that are used for actually culturing cells. And if you were thinking about cultured flask, there are little tea flasks that often is used in the laboratories. They can only offer only to a certain limit of number of cells. Here you can see on the graph roughly about 5 to 10 billion cells. And that's the maximum kind of you can achieve. And then it's not really it's a matter of footprint. You can't keep stacking them. It's not very ergonomic to actually get cells in them and detach them and recover them and so on. So the next bit of the kind of the technology that was developed was originally cell factories. They are essentially layered flasks in different layers. They can go up to really large number of layers, but of typically 5 to 10, you can see. And still, despite the fact that they might be a very good, easy to use plastic, but still they are bulky, hard to use. It's very laborious, heavy, and then imagine if you were to actually use a lot of this, it's still very economical at large scale when you actually need lots of cells. You're talking about trillions of cells. So that's where bioreactors often come in, that you need to scale it to bioreactors, and particularly for adherent cells, there's no other option than really using macrocarriers. There are other technologies like hollow fibers or packed bed bioreactors that use other techniques for growing adherent cells, but still, we believe at least one of the most scalable methods has been use of macrocarriers within bioreactors. Now, when you think about the therapy, it is similar in the sense that you need a lot of stem cells to actually inject to the patients, and that's one of the key components in the cultured meat space, as well as the cell therapy. Now, to give you some perspective actually on how it starts, so cells, when you want to grow cells in the macrocarrier, it involves of 3 main steps. The first one is cell seeding on the carriers, where essentially cells will be seeded and they find these sites of the macrocarriers, the surface of them, as a seeding site. So they can actually go and attach, and they simply start attaching, start elongating and proliferating. So they can expand in number, they can grow to the maximum number required. And after you got the number of cells you like, then you got to often detach these cells. So in the conventional carriers, you have to go through the process of detaching them with an enzyme similar to the flasks. And then you get these large macrocarriers from little cells. So macrocarriers roughly 150 micron in size, small little cells, 10 to 15 micron or so. And then you can simply filter them out and get rid of the kind of the carriers, so you're left with your cells. Essentially, if you were kind of to put this in a visual perspective, right? The conventional flask or tea flask that we've been speaking about, this is actually to culture 250 ml. You can see here we are holding 2 identical bottles next to each other, so you can have an actual visual perspective of what it is to culture around 120 million cells. And obviously, macrocarriers can make things in much more less space, like 26 times less space and 425 times less cost compared to the flasks. And what this means is essentially is you can enable really the scale up. You can really go beyond, and you can go to thousands of liters easily. And the idea is that, this is the way to go forward for culture meat if you are to make adherence on a large scale. Now, Conventionally, microcarriers have been really problematic to use, and the first and the foremost problem, especially for the cultured meat space, is the use of non-biodegradable materials like plastic or glass that can end up in the final product. They can break throughout the culture, they can leach out, and simply they're not eatable. So it's definitely one of the biggest concerns and points of kind of challenges for the cultured meat to use microcarriers that were in the market originally. The second problem is the 50% cell loss that you have due to the workflow inefficiencies. So essentially, after growing your cells to large scale, you can lose 50% of them in those two steps, particularly of detachment, of detaching the cells from the microcarriers, and the step of actually filtering the carriers away. And then the next one is really costly. So it can really cost over a few $1,000s for just culturing 1 billion cells. And when you put that into perspective of making a kilogram of meat, that's really, really not a financially viable option. So essentially, to overcome these problems, we have developed the next generation microcarriers made from natural biopolymers that are completely eatable, and they're already safe materials that have been proved and used widely in the food sector. So we don't use any harsh chemicals throughout the production, and that's what makes our product kind of very unique. We eliminate the cell loss as well by eliminating the attachment and filtration steps. So often our microcarriers can either be used as the eatable scaffold directly for the final product, or it can simply just be digested away using another enzyme or non-enzymatic buffers. So that perspective can really improve the whole workflow and yield to much higher cell yields essentially. And the next thing is we can make this affordable, so it can cost only a fraction of what is already out there. And most importantly, our microcarriers can be a component of that, that scale up of the cultured meat. So it can only cost only a fraction of the final product, rather than being it the other way, right? So that has been the core part of the company, and our product features include first use of flexible materials, so we can have serum-free, biodegradable, and eatable options. So these carriers can really have different types of materials in them. They can be really affordable, so you can enable that scale up without sacrificing the economical viability of it. You can simplify the entire workflow by eliminating those wasteful processes and get some kind of a better sort of cell yield as well. And finally, customizing it. Now, making them cell type specific. And what I mean by that and why that matters is essentially, let's look at the simple example of a stiffness of different kind of organs in the body and the different tissues. So you can see here along different sides, for example, your liver has different Young's modulus and stiffness compared to your muscles or compared to your bones. So that part is what we are missing in the whole process when we are growing cells in the lab. So when we want to grow stem cells, stiffness is a really critical factor to mimic actually the microenvironment in the body. And our microcarriers can be customized so that they can be exactly precisely with a certain stiffness. So cells can simply feel and mimic the same way that they were to grow inside that animal body. So we can really fine tune them, particularly most of the cases you're talking about 3 to 12 kilopascals for muscle cells, but you can really go beyond and have really higher stiffness cells and higher stiffness microcarriers along. So now this is only one of the aspects that is the stiffness. Now, if you were to put that in perspective with a bunch of other different factors like size, charge of the microcarriers, functional groups, matrix composition, morphology, shape, and surface coatings. All of these are the other things that we can fine tune. And we believe that they play a huge, huge role in actually getting the best cell results. And that's what has been missing in the entire microcarrier history that has been around for over 50 years maybe, but still we cannot really customize them in terms of all these little physical or chemical features. And they play a critical role in this whole process. And on that end, we can really fine tune this from different materials, bunch of different hydrocolloids. We can really mix and match different things, and we can have things from very soft and flexible materials all the way to the really firm and brittle. But the key part is we can really fine tune them and find the microcarrier texture that is required for the application. And now, especially, this would be very critical for applications that you are actually involved with direct scaffolding of the carriers for the final meat product, so you can make that mouth feel the way that the client essentially needs. Now let's dive into a bit of experimental results. So here's some results showing that myoblast cultures on our carriers, within just simply 6 days, we can achieve over 500,000 cell densities without the change of media and direct culture essentially, and we can achieve over 30-fold increase in terms of the expansion of the cell. The cell attachment is often pretty good. Over 90% attachment can happen within the first 6 hours. And that really helps and facilitates that growth. And then you can compare that with the commercially available carriers in the market that it can yield to much higher cells. And to give you some more visual perspective, here's some pictures of actually what cells look like on day 0, where roughly 1 to 4 cells are attached per bead. And within 6 days, you're essentially having the entire surface of the microcarriers grown and confluent with cells. So some more cool SEM images that I thought might be interest of you. So it's actually showing that how cells can elongate on the microcarrier, and these are particularly muscle cells elongated. And we have more pictures of the SEMs of different type of fibroblasts here growing on different type of carriers. And then here are some more experimental results on the fibroblast end. So fibroblasts, they can yield to much higher cell densities, 1.5 million per ml within 8 days that we got on 125-fold increase. Essentially, again, you can see clear difference with products in the market, that how our products, how our microcarrier can actually outcome them and actually yield to much higher cell yield. And note that we customize these carriers for each of these cell types. So this is kind of the best of all matches in terms of the stiffness of the carriers, the condition, size, porosity, and so on. So you can see here how fibroblasts cover actually our P1 microcarrier, particularly before and after the cell coverage. And we've also been using these carriers for other applications, including some virus host cells for making viral vaccines or viral vectors. And also similarly with mesenchymal stem cells, where we actually achieve 600,000 densities within 5 days of culture simply. You can have a further closer look with the stem cells here, how they grow very nicely in kind of like a monolayer culture that we are aiming. And we're showing that really we are not changing any of the markers of these or the stemness of these cells across different markers like CD34, CD45 as the negative markers, and positive markers of CD90, CD73, and CD105. Now, giving a more of like a head-to-head comparison in visual perspective, like a simple HiMedia cytometer, when you dissolve the microcarriers, you can simply see how much higher cell yield we can achieve. And similarly, you can compare them with other digestible carriers that within the same time we can actually completely digest the carriers and get all the cell yield that required. Often the carriers, depending on the case and the type of carrier, we can digest them anywhere between 5 to 20 minutes. In this case, is a stiffer carrier that will take 20 minutes of digestion, and you can see how the before the when we harvested the cells in a Falcon tube, you can see the carriers with yellow media, and simply after digesting them with enzyme, they simply just disappeared, and you have your cells ready to centrifuge and take on for the next step. Now, some more kind of visual microscopic images. In day 1, you can see some live cells on each carrier. 1 to 4 cells, again, is the typical range that we often grow cells on. And within the 5 days here, you can see complete confluency, and this is already directly suitable for direct scaffolding. Or in some cases, you will want to recover the cells. We can simply just harvest the cells and simply digest them and achieve all your cells after that. Now, some commercial microcarriers have really big issues that they are overcoming. So one part is the detachment issues with cells, and that's particularly one of the reasons, as I mentioned, that will cause the cell loss throughout the process. And that is where cells actually don't come off from the carriers. And you can see here, cells detach when you treat them with the enzyme, but a lot of them actually remain on the carriers. You can see the elongated cells here. And that's one of the biggest problems that is really a challenge in the microcarrier culture. But also is also also fractured macrocarriers that they can break throughout the culture. So now that will bring a lot of concerns around residuals and particulates of these carriers inside the final product that can come out. So that's why we really think the either digestible macrocarriers that can fully digest or eatable ones that you can directly scaffold them is a critical thing in this sector. And in the detachment end, you can completely see here we are comparing just the detachment efficiency, and there is a clear difference between, for example, here Cytodex3 that we have actually done a head-to-head comparison, and you can see there is a clear difference in number of cells that you can recover from these carriers. Because often Cytodex obviously is not digestible, and you need to go through that filtration step, and that causes a lot of problems. And similarly, more pictures after the filtration, you can see how that's completely different in terms of cell densities that you get. So we similarly compared these carriers with commercially available carriers, and these are some of the benchmarks that we use. And really the whole gold standard is really what we are actually trying to get is digesting the carriers fully is a critical matter if you're trying to recover the cells, and that is a key kind of advantage of our product. Now heading to the second set of products in the smart MCs, which is our hydrogels. So the smart hydrogels really come in is more on the proof of concept stage, and we are looking to launch this product later this year. And they're used for mimicking the tissue in the laboratory for a range of different applications. And the products really come in with the xeno-free and fully defined metrics that we can really use them, that you know exactly, unlike collagen that has been often used in the field, you can actually use this as a fully defined metrics and actually have reproducible results every time within your culture. The whole process is one-step operation, so you simply load your cells inside these gels, and then the gelation can start happening within a given time frame, like 10 minutes. So you have the time to inject them into your device as well, plates or wherever you're looking to actually mimic that structure. Their temperature is stable, so we can have them at room temperature, and they're stable during the culture as well. So, and also they will have a simple release option where you can simply release the cells without any enzyme and with an enzyme-free essentially buffer. And this release mechanism can let you recover the cells essentially. So some examples that we have been doing some research on has been brain organoids, tumor spheroids, and ECM hydrogels. But essentially, we're looking actually to expand these, and especially I think that will be a very cool point of research also for a lot of culture meat researchers. Now down to the third kind of set of products, and that's our microfluidic solutions. They range really from different type of cartridges. So a lot of these are used in R&D scale, but some of them, which I will talk more in the next slides, also can be used really in the bioprocessing stage. But this might be of your interest, especially for your researchers at academic or literally at an industrial scale. So we have a bunch of different products around cell sorting. So you can size, based on the size of the cells, you can sort the cells from different biological samples. And these cell sortings have been really used as a label-free method to actually separate different sizes in a high throughput manner and clog-free. And that's a key component that unlike the filters or other methods for separation, they provide these benefits. Smart tubes to make actually effortless droplet generation on the spot, followed by droplet microfluidics. So if you're looking to actually generate droplets, we can help you actually build those devices for you, or actually help you, educate you on how to operate them. And that can be one of the really cool points of research, particularly in the culture meat space. Microware nanowells, scratch assays for cell patterning, and microfabrications, other services that we provide to the clients. And really, if you were interested in more talks on microfluidics, we are really experts in the field, and we can provide you with a lot of help on what microfluidics can bring you, or what limitations it might have for your applications. And some visual pictures of like cases here is a cell sorting, where you're sorting bigger cells from smaller cells in the device. Plasma separation, scratch assays like cell patterning that I mentioned earlier, and static droplets that you can make static droplets on the device for different co-culturing or analysis of cells, even down to single cell resolution. Now, one key products that might be of very interest to this to the cultured meat space might be the cell harvester that we are developing. So this product, essentially, the idea is it can, in a high throughput manner, clog-free manner, it can harvest your cells from your bioreactor sample. So imagine a bioreactor that is running. You can passively simply pump the liquid out through this device that uses the microfluidic solution to separate your waste media from your cells, and they will concentrate your cells essentially. That can be fed back to the actual bioreactor for in a perfusion manner, or it can go to another collection essentially tube, and each of those modes can provide an important aspect. So if I would to provide you with like a bit of context on why microfluidics can bring some benefits here, you can see the efficiency and throughput in different types of ways of actually using microfluidic solutions. So a lot of systems like sedimentation, centrifugations, they often have a limitation on one side is throughput, and the other side is actually the requirement on the equipment. But these work that we call them integrated microfluidics can actually bring you the benefits of actually higher throughput and a better efficiency. So without the need actually to rely on really like sophisticated methods like centrifugation, you can actually dewater your samples. And I'll give you some context, and this is how the device actually works under the microscope. So we can actually separate your cells from all the liquid simply by running this device. And some more visual context here is, for example, here is if you were to actually think about a 50 ml liquid here, with a high concentration of cell, we can essentially bleed 80% of the liquid out and have you with it only 20% of the solution. Now, what this means is, one thing is you can harvest the cell at really high throughput without any problems like clogging, like filtration would bring. But the other side of thing is, for example, this would be an excellent thing if you were to actually do this to reduce your total volume for centrifugation. So as a pre-centrifugation step, you can simply reduce your volume of your liquid. For example, imagine of a 1 liter culture might be more difficult to centrifuge, while 200 ml can be much simpler to process. So that's kind of the mentality. Or, as I mentioned earlier, it can be a passive perfusion of your bioreactors for bleeding the bioreactor and removing the waste media and adding cells back to the bioreactor. So these are the kind of, this product is something that we are really interested in working with different companies to further developing it, but also to suit it to your needs actually. So of course, this will be much more, application specific type solution. So if you are very interested, I'll be very keen to kind of speak with you, and our team can really provide you with all the context on that end. So again, one more kind of pictures on the how it looks in terms of like the original sample, where you can concentrate them and you lose almost no cells in the whole process. So this is one of the key components of this whole process. Now, to kind of sum up the whole thing, I know I've been talking quite a lot. I'm sorry if I'm speaking a little bit too fast, but the recording of this video hopefully will be available on GFI YouTube website. So in case I spoke too fast, or you wanted to have a quick review of the of this presentation, please go to that GFI YouTube website, and or reach out to us directly, and we can answer any questions you might have. But essentially, the whole process, our products can break into those 3 parts of the microcarriers, which we have 3 main category of carriers with many subtypes and customization down there. We have the hydrogels coming in and different microfluidic cartridges that can provide you with different aspects on the actually your research or your industrial process. So thank you so much for the time for listening to me. And here's our details. Feel free to scan the QR code here, or directly reach out to us via our email. That's probably the best way you can reach out to us, or on our LinkedIn. We often post different news of our company, or different kind of cool, interesting findings in the microcarrier space and hydrogel and biomaterials. So yeah, I'm really pleased that I had the opportunity today to give this presentation to you, and really appreciate any comments or feedbacks you may have in this sector, and any points of points of collaboration that you think we can work together. I'll be keen to hear more from you guys, and thank you so much. Bye-bye.
Well, thanks to everyone who stayed for the extra long meeting this month. A lot of great information from from HiMedia and Smart MCs. As Payer said, he's in Australia, so he's not here to answer questions live. So if you do have any additional information you'd like, you can reach out to him directly. And we look forward to seeing you again next month. And you'll be receiving the follow-up email with the full recording and contact information for our presenters. So thanks again for being here, and we'll see you again soon.